Hello, fellow warriors. Here we are with news from the website prostatewarriors.com.
Thank you. It is really great to be diving into this data with you today.
Imagine finding out that the very defense mechanisms your cancer uses to survive and spread can actually be hacked, like reprogrammed and completely stripped away. Welcome to this deep dive.
It is a critical time to be looking at this, honestly. The scientific community has largely moved past the era of simply throwing broad, highly toxic treatments at the wall just to see what sticks.
Right. And if you're joining us, you already know that navigating an advanced prostate cancer diagnosis can feel incredibly overwhelming, especially when the medical field sounds like it's speaking a completely different language.
Exactly. The jargon alone can be a barrier.
So our mission today is to cut right through that noise. We are exploring the newest, most promising treatment strategies for advanced prostate cancer. We're going to focus first on the active clinical trials happening right now before we look at some really fascinating early-stage lab research.
And what we are seeing now in the clinic are highly targeted, strategic counterattacks based on the specific biology of the tumor you are fighting.
Okay, let's unpack this. We should start with the big guns, which means the phase 3 trial.
Right, the massive late-stage studies.
Exactly. These are the ones actively pushing the boundaries of standard care for aggressive disease types. And one that really caught my eye is the CaBRA trial. It is testing a quadruple intensification therapy for metastatic castration-sensitive prostate cancer.
Just the word quadruple makes it sound incredibly heavy, I know.
Yeah, it does.
But it is an aggressive escalation that is highly calculated. The current baseline for these patients typically involves a docetaxel-based chemotherapy backbone combined with lifelong androgen deprivation therapy, or ADT, and a drug called Darolutamide to block the androgen receptors.
Right, so that's 3 things already.
Exactly. In the CaBRA trial, they're introducing a fourth agent into the mix, which is Carboplatin.
Right.
But they're only doing this for tumors that show specific vulnerabilities.
Primarily BRCA alterations or neuroendocrine differentiation, right? I want to break down why that specific combination matters, starting with the BRCA alterations.
Sure.
Because we hear about BRCA a lot with breast cancer, but for prostate cancer, a BRCA mutation essentially means the cell has a broken homologous recombination repair mechanism.
Which sounds like a mouthful.
Yeah, a huge mouthful. But if I understand it right, it basically means the cancer cell has lost its ability to accurately repair its own DNA when it gets damaged.
Precisely. The cell essentially loses its primary spell checker. When a normal cell replicates, it fixes errors in its DNA. A BRCA-mutated cancer cell can't do that efficiently, which, allows it to mutate and grow rapidly.
But that broken machinery is also its Achilles heel.
Exactly. Because carboplatin is a platinum-based chemotherapy, its entire job is to cross-link the DNA strands so they cannot unzip and replicate.
Oh, wow.
Because the BRCA-mutated cancer cell is already terrible at fixing DNA damage, hitting it with platinum causes catastrophic, unrepairable damage to the tumor cell. It leads straight to cell death.
So you are basically taking the tumor's broken machinery and using it as a weapon against itself. But the CaBRA trial also targets neuroendocrine differentiation, and that is a totally different beast, right?
Radically different. Neuroendocrine prostate cancer is a shapeshifter. It stops relying on the androgen receptor to grow, which means standard hormone therapies suddenly become useless.
Wow. And it often stops producing PSA as well, doesn't it?
It does, making it much harder to track. Because it behaves so aggressively, introducing a heavy hitter like platinum chemotherapy early in the treatment sequence is a necessary escalation to head off that rapid growth.
It makes complete sense to match the weapon to the specific armor the cancer is wearing. But what happens when the cancer survives the early lines of defense?
That brings us to the VESITRE-01 trial.
Yes, another massive phase 3 study enrolling about 670 participants globally. This one evaluates an alpha-emitting radioligand therapy called AZD2265, or, if we want to get incredibly technical, 225-AC PSMA-INT.
The patient population for VESITRE-01 is really what makes it so critical.
Because it's for patients with PSMA-positive metastatic castration-resistant prostate cancer, or mCRPC.
Right. But specifically, this trial is for patients who have already been treated with and progress on beta-emitting therapies like 177-Lu PSMA 617.
Which many listeners might know as Pluvicto. I really want to visualize the difference between beta and alpha emitters for a second, because the physics here are wild.
The physics are fascinating, yeah.
Think of beta emitters, like 177-Lu PSMA 617, as a broad floodlight. When the radioactive isotope attaches to the cancer cell, it emits radiation that covers a relatively wide area. It penetrates deeply, but the energy is a bit more diffuse.
Right, like a floodlight.
But an alpha emitter, like the AZD2265 used in this trial, is like a high-powered, short-range laser. Alpha particles are massive compared to beta particles. They travel a very, very short distance, sometimes just a couple of cell widths, but they deliver a devastatingly high amount of destructive energy.
The physical impact on the cell is profound. Validate that analogy completely. A beta particle might cause single-strand DNA breaks, which the cancer cell can sometimes survive and repair.
But the alpha particle.
An alpha particle, because of its sheer mass and energy, bulldozes right through the DNA, causing double-strand breaks. The cell cannot recover from that.
Wow.
So if a patient's tumor has stopped responding to the floodlight of a beta emitter, hitting those same PSMA-positive cancer cells with a heavy laser of an alpha emitter offers a completely different mechanism of damage.
And it is worth noting that the VSTORY-01 trial explicitly excludes patients who have had prior alpha emitter therapy, with the one exception being radium-223.
Correct.
If this trial hits its endpoints, could this drug become the first approved alpha emitter for this specific post-Pluvicto indication?
Yes, and that would fundamentally rewrite the rulebook for sequencing treatments in advanced disease. Right now, patients failing beta emitters have limited options, often looking at cabazitaxel chemotherapy or switching hormone agents. A targeted alpha therapy offers a vital new lifeline.
Since we are talking about patients running out of standard options, I want to pivot to a historically difficult challenge. We're looking at 2 phase 2 trials focusing on patients with visceral metastases, meaning the cancer has spread to organs like the liver or soft tissue metastases.
Yes, historically a very difficult population to treat.
But before we get into the trial data, I have to ask, why are visceral metastases, like liver involvement, historically so notoriously difficult to treat compared to bone metastases?
It has everything to do with the environment of the organ itself, particularly the liver. The liver is the body's primary filtration system. It is constantly processing toxins and foreign material from our blood.
Okay.
To do that without triggering massive life-threatening inflammation every day, the liver has a highly specialized immunosuppressive microenvironment.
So it naturally dampens the immune system.
Exactly. When prostate cancer cells land in the liver, they hijack this tolerant environment. They essentially build a protective fortress that shields them from the body's immune system and makes them highly resistant to standard hormone therapies and chemotherapies.
So the cancer is literally using the liver's natural peacekeepers to hide. That explains why the YL201 phase 2 trial is turning so many heads.
Right.
YL201 is an antibody-drug conjugate, or ADC, targeting a protein called B7H3. They tested this in 82 heavily pretreated patients with advanced mCRPC.
And when we say heavily pretreated, the median was 4 prior lines of therapy. Over 70% had prior taxane exposure.
And almost half of them had visceral metastases.
Patients in that demographic traditionally face very poor prognosis. An antibody-drug conjugate is essentially a smart bomb. The antibody acts as the guidance system seeking out the B7H3 protein on the tumor.
And once it attaches,
it delivers a highly toxic drug payload directly into the cell, bypassing a lot of those liver-specific resistance mechanisms entirely.
And the numbers back that up. They saw a confirmed PSA 50 response in 38.5% of the group. But even more critically, the overall response rate, or ORR, which measures actual physical tumor shrinkage, was 29.5%.
Those are impressive numbers for that stage.
Yeah, and the median radiographic progression-free survival, or rPFS, was 9.1 months. And it gets better. When they look specifically at the 2.4 milligram per kilogram dose, the PSA responses hit nearly 49%, and that rPFS extended to 11.8 months.
Let's contextualize that 11.8 months. For a patient with visceral metastases who has already exhausted 4 prior lines of treatment, gaining nearly a year where the cancer is not radiographically growing is a massive clinical achievement.
Truly massive.
It shows that targeting B7H3 works even when the tumor has outsmarted everything else.
I noticed the safety profile was considered manageable, primarily involving neutropenia and anemia. But, for a listener facing this, manageable still means side effects.
Yeah.
What do neutropenia and anemia actually look like for a patient day to day?
It relates to bone marrow suppression. Neutropenia means a drop in neutrophils, a type of white blood cell which significantly increases the risk of infection. Anemia is a drop in red blood cells, leading to profound fatigue and shortness of breath. While these take a serious toll on quality of life, they are considered manageable by oncologists because they can often be supported with growth factor shots or transfusions.
Instead of causing irreversible organ damage.
Exactly.
Okay, here's where it gets really interesting. There is another phase 2 trial tackling these tough visceral and soft tissue cases called the Zanzalintinib trial. It's enrolling 19 participants with mCRPC who have already progressed on an androgen receptor pathway inhibitor, or ARPI.
Yes, Zanzalintin.
The drug zanzalintinib blocks the tumor's blood supply through VEGFR2 inhibition, but it also blocks something called the MET and AXL MERTK pathways. I'm going to need a translation on that alphabet soup. What are these pathways doing?
VEGFR2 inhibition is fairly straightforward. It starves the tumor by preventing it from growing new blood vessels. But tumors are incredibly adaptive.
They always find a way.
Right. When you cut off their blood supply or block their androgen receptors, they open up alternate molecular pathways to survive. MET and AXL MERTK are essentially the escape hatches the tumor uses to keep growing. Zanzalintinib slams those escape hatches shut. What's fascinating here is the secondary effect this has. Prostate tumors are typically immunologically cold, meaning the immune system just glides right past them without recognizing a threat.
But by shutting down those specific escape pathways...
Zanzalintinib physically alters the tumor microenvironment, converting it from cold to hot. It strips away the tumor's camouflage, uncloaking it so the body's immune cells can actually see the danger.
Uncloaking it. So it paves the way for immunotherapies to finally work on prostate cancer.
Precisely.
Okay, but this raises a fundamental problem. If tumors are constantly adapting, building fortresses in the liver, or opening up new escape hatches, how do we stop them from adapting in the first place?
That is the big question.
Right, which leads directly to trials that are targeting the cancer software to reverse this resistance entirely. We are talking about epigenetic reprogramming.
Epigenetics is arguably the most exciting frontier in oncology right now. You are essentially trying to reprogram the cancer cell's memory. Let's look at the KAT6 inhibitor phase 1-2 trial.
Okay.
It is enrolling about 36 patients, combining an investigational KT6 inhibitor with the standard drug darolutamide in mCRPC.
What exactly is KKT6 doing inside the cell that we need to inhibit it?
To understand KD6, we have to talk about chromatin accessibility. Think of the DNA inside a cell as millions of highly detailed blueprints.
Okay, blueprint.
To fit inside the cell nucleus, those blueprints are spooled tightly around proteins called histones, like thread around a spool. KT6 is an enzyme that unspools specific sections of that DNA, making the blueprints accessible so the cell can read them.
I see.
In prostate cancer, KAT6 unspools the blueprints for oncogenes, the genes that drive cancer growth, like MYC, keeping them turned on.
Wait, let me try an analogy here to make sure I'm grasping this. Think of the cancer like a computer system that has been hacked by a virus.
I like that.
Our standard targeted therapies, like our ARPIs, are the antivirus software trying to locate and delete the malicious files. But the hacker has gone deeper. They have rewritten the computer's underlying operating system to completely ignore the antivirus.
Yes.
That operating system is the epigenetics. The KAT6 inhibitor is basically redisciplining the hacked computer. You aren't just smashing the hardware, you are resetting the corrupt software, spooling those bad blueprints back up and locking them away, so your standard antivirus can actually work again. So what does this all mean?
It means restoring a tumor's vulnerability.
Yeah.
When you give darolutamide, you are directly blocking the androgen receptor. But the cancer often uses its epigenetic software to bypass that block.
By adding the KAT6 inhibitor, you disable the software, allowing that bypass.
Exactly. You are trapping the cancer in a corner it can't mutate its way out of. Create a powerful two-pronged approach.
We are seeing this exact philosophy in another trial, the MOMENT trial. This is a phase 2 study with 60 men testing another epigenetic drug. This one is an EZH2 inhibitor called mevrometostat, and it's combined with enzalutamide.
Yes, the MOMENT trial.
But what strikes me about the MOMENT trial is the incredibly specific window of patients they are looking for.
The enrollment criteria are very rigid. They are targeting patients who have progressed on an earlier ARPI like enzalutamide, darolutamide, or apalutamide, but who have not yet progressed on abiraterone.
That is a very narrow slice of the treatment timeline. Why mandate that specific sequence?
Because of where it sits in the sequence, it is about maximizing the patient's long-term arsenal. Often patients get an ARPI early on. When the cancer eventually figures out a workaround and becomes castration-resistant, oncologists have to decide what card to play next.
Right.
By stepping in at this precise moment with mevrometostat to rediscipline the epigenetic program, They are trying to squeeze more time out of the ARPI pathway.
Squeezing every last drop.
Exactly. Crucially, they are doing this while keeping other proven standard options, like abiraterone, safely in the back pocket for later. You never want to burn a bridge to a viable treatment before you have to.
Maximizing the mileage of every single line of therapy. That is exactly what you want your medical team doing. All right, we've covered the heavy-hitting Phase 3 and Phase 2 clinical trials. Let's zoom out and look into the future, specifically at the early-stage preclinical pipeline.
The lab research.
Yes. If we go back to the CaBRA trial at the beginning of the show, we talked about that highly aggressive neuroendocrine phenotype, the shapeshifter. Scientists are currently in the lab trying to figure out how to stop that transformation at its biological root.
And they have zeroed in on a key driver, which is a protein called SRT1. In preclinical models, researchers discovered that SRT1 isn't just a passive marker. It is the active engine pushing regular prostate cancer cells into that dangerous, therapy-resistant neuroendocrine state.
And when they inhibited SRT1 in mouse and cell models, the tumor growth slowed down, and those aggressive neuroendocrine features were noticeably reduced.
Right.
They're looking at a compound called salicystat to block it. But I want to play devil's advocate for a second.
Go for it.
Since this is just happening in mice and cell models right now, why should a patient listening to this care about these early findings today? It feels like it could be a decade away.
It is a completely fair skepticism. The leap from a mouse model to a human clinical trial is notoriously difficult. However, the reason this specific finding matters today is the compound itself.
Salicystat.
Yes, salicystat. This isn't the hypothetical chemical they have to invent from scratch.
Mm-hmm.
Salicystat already exists.
Yeah.
Because researchers have identified an existing compound that hits this target, it creates a drug repurposing path.
Oh, wow.
They bypass years of initial safety testing and molecular engineering. Finding a repurposing target significantly accelerates the timeline from the lab bench to human trials, offering tangible hope that the pipeline for these lethal variants is expanding rapidly.
That dramatically shortens the runway. Okay, we have covered a massive amount of ground today. We started with the sheer force of Phase 3 trials, looking at the CaBRA trial's quadruple therapy for vulnerable tumors.
And Vedra A Year 1, using highly targeted alpha radiation to destroy cells that have survived standard beta emitters.
We tackled the toughest visceral metastases with the YL201 antibody drug conjugate and Zanzalintinib's ability to uncloak tumors. We explored the mind-bending science of the KT6 and MOMENT trials, literally resetting the cancer's epigenetic software to make it vulnerable again.
And finally, we saw how preclinical work on SRT1 is aiming to stop the deadliest neuroendocrine transformations before they take hold.
Yeah. The overarching takeaway for you, the listener, should be one of profound optimism regarding the strategy of modern oncology. We are witnessing a fundamental shift away from blunt force treatments toward precision biological warfare.
Exactly. The medical community is learning how to exploit the cancer's broken DNA repair, shut down its escape hatches, and even rewrite its epigenetic memory. These treatments are becoming as individualized as the patients receiving them.
It's an incredible shift in momentum. We are finally dictating the terms of the fight, rather than just reacting.
And I'll leave you with a final thought to ponder. If we can use drugs to successfully reprogram a tumor's epigenetics, making it vulnerable again, could we eventually reach a point where cancer is treated not as a disease to be eradicated, but as a chronic condition that we constantly outsmart and reprogram indefinitely?
Wow. Reprogramming cancer into a manageable chronic condition, that is a future worth fighting for. And based on what we've unpacked today, it feels closer than ever. Thank you for joining us on this deep dive. Stay strong and fight on.